So here is the neuroblast, and when the cells move up and down, they will actually detect in their environment that at that layer, they will look at the signals, and those signals will tell that cell what to become. And then they will differentiate into a particular neuron. And this is really one of the things that allows us to take stem cells and squirt them into the brain, and those stem cells will go in and actually look around, identify what area of the brain it is, and then become a cell that's in that part. So this is what we're doing with treating of Parkinson's, where we know that the substantia nigra has died off, all these dopaminergic cells are gone. We can take stem cells and squirt them into substantia nigra. Those stem cells just look around, say, oh, here are the clues that tell me what cell I can be, should be, and become those cells. So once the cells have sort of identified themselves, then they go send out axons and dendrites. And so the axons are, at the tip of the axon is what's called a growth cone. And these guys actually go out and move around and sort of crawl through the brain to find some kind of target to synapse onto. And this is exactly what Dave Odie studies, is how do these guys form, how do they move, how does collagen or the microtubules create these guys, and how they signal so that you know this guy is the one we want to move along and this one is not. He's also found that the rate of the Lipodia movement, the growth cone movement, depends on how stiff the substrate is, so you can optimize the rate of growth by changing, you know, how stiff the gel is that you're growing the cells on. So when early on there are spots like this cell. So here's a cell that needs to cross over to the other side of the spinal cord, for example. So it was born, here, migrated out here, ends up sitting there, and now it's got to send out its growth cone. This growth cone has all sorts of receptors on it, and these receptors start looking for a couple different signals, and one of them is netrin, which is a chemoattractant. It is a molecule which is being released from the midline of the animal and is telling cells to come here. And it goes up the gradient and finds, and once it gets to a high enough concentration, the gradient, it actually stops producing the netrin receptors and starts producing receptors for SLIT. And SLIT is another protein which actually tells the cells to go away. And so they come along, they come here when they got this, I guess some momentum keeps going in the same direction, they turn on these things which push them out to the other side. Does that make sense? So as it's coming in, so what's being produced here at the midline doesn't really change. What happens is the cell changes its receptors from ones that detect things that pull it to ones that push it. So then it goes up, and maybe it's found its target. Finding its target is a really complex thing. I mean, can you imagine crawling through the body and trying to figure out where you're supposed to go? I mean, there's just so much, but once it gets there, it goes and identifies that there's another axon, and it actually sends out, the dendrite sends out what's called a filopodium, and it's a little finger that comes out, it touches it, and that touch actually triggers the axon to actually start producing synaptic vesicles and a presynaptic area. So this is a push-pull system. It sends out, touches it first, that actually makes this guy start to differentiate that membrane and produce the synapse, which then starts to trigger the postsynaptic cell to start forming receptors. So here you start seeing the receptors start forming. So even though this guy triggered the connection first, it really waited until it saw the response from the presynaptic cell before forming a postsynaptic specialization. So we go out and we send out lots and lots of synapses initially. We don't know what synapses are good and what synapses are bad, so we make lots of synapses initially. And then what we do is we cut them back and we refine the whole system and clean it up. And so we might have lots of input neurons to start, coming to some target neurons. Some of them are actually connecting to guys there, and some of them aren't. And these guys actually will send out a little signal to say, okay, you're connected to me, you're good, you stay alive. And if another guy next to you doesn't actually connect to anybody, he doesn't get that signal, and he dies off. And so eventually you get only the guys who actually send connections surviving. And these guys who didn't actually connect actually die through a really elegant process where they actually sort of decompose themselves through a process called apoptosis. Initially, there's lots and lots of birth of cells, and then even in utero, there starts lots of, and I'll show you some pictures of how it's even being refined in utero. Dogma was that there are no real new neurons being born shortly after birth or before birth, and everything's just refinement after that. But there's now evidence that there are subtrabecular zones which are still producing cells throughout, and a lot of them are replacing olfactory neurons and stuff like that. And there's stuff that's going into the hippocampus too, and rewiring. It can be triggered after epilepsy, and those are really interesting things. But there's an astounding number of neurons that are dying per day, on the order of like 50 to 100,000 neurons. I forget the details, but we have a lot of neurons, so it's a small fraction that's dying every day, but the numbers are astounding.